Hello, Biochemistry Simplified, Madam Winnie here. I thank you all for subscription. So keep on subscribing, everyone, so that we can keep on learning. But I thank you. Thank you so much, and tell your friends we keep on subscribing. And I hope at the end of the day, the channel will be of help to students. So after diving into amino acids, I just have a short lecture on peptides. Just as we were talking about glycosidic bond involves the carbohydrates, a peptide bond is also involved in proteins. The difference between a peptide bond and a glycosidic bond is a peptide bond is formed in proteins, glycosidic bond in carbohydrates, but they have a similarity. Peptide bond and glycosidic bond, a water molecule is released at the point we are forming a peptide bond. So if you talk about a dipeptide, a tripeptide, a tetrapeptide, the di, tri, and tetra speaks much about the number of amino acids involved in the bond formation, not the number of bonds forming, the number of amino acid residues involved in the formation of the bond. So if you talk about the dipeptide, 2 amino acids are involved, tripeptide, 3 amino acids are involved, and tetrapeptide involved 4 amino acids. In each and every point where a peptide bond is forming, a water molecule is released. So if you talk about a peptide bond, then we're talking about the proteins. Now we're already forming the protein because it will be a polymer of those amino acid residue attached in a linear order. So that is how the primary structure of a protein looks like. All the 20 amino acids with a peptide bond in between. So if you talk about a peptide bond, you're talking about the carboxyl terminal end of one amino acid residue connecting with the amino terminal end of another amino acid residue with the release of water. The carboxyl and the amino attaching themselves, but then there is water released. So then that is how a peptide bond forms. So you talk about a peptide, it consists of 2 or more amino acid residues which are linked by a peptide bond. But then it forms when the carboxyl group of one amino acid reacts with the amino group of the other amino acid, and even at that point the 2 amino acids are combined from the carboxyl and the amino terminal end, forming a peptide bond. A water molecule is released. So you further talk much about the amino terminus, or the end terminus of an amino acid, which is always at the left, and the carboxyl terminus of another amino acid. So in that case, we'll be able to release a molecule of water. So a peptide or a protein is named starting with the end terminal. The end terminal is on the left hand side, and then you usually read it as you go to the carboxyl terminal end. So usually the end terminal is located to the left hand side. So what shows here is how a peptide bond looks like. You have the amino terminus where you can see the nitrogen, hydrogen, and hydrogen. I told you balancing the bonds around the nitrogen. Bear in mind that it is atomic number 7, 2 to 5 configuration. The outer shell would need 3 electrons for stability. We have the central carbon here, we have the hydrogen, and we have the carboxyl terminal end. This is the R side chain, carboxyl, amino. Carboxyl terminal end of the first residue, amino terminal end of the second residue. A peptide bond is forming. At this point, you can see the OH and H name in red. A water molecule is released. That is how a peptide bond forms. So I said we can talk about a dipeptide, you can talk about a tripeptide, you can talk about a tetrapeptide, which further we discuss a polypeptide. Poly, you can't even count the number of amino acids involved in the bonding, and that is when it forms the protein. So you can't talk about a protein without talking about the amino acid. Without the amino acid, the proteins can't exist. So that's why we had to start with amino acids. So if you talk about a dipeptide, it involves 2 amino acid residues and a 1 peptide bond is forming, meaning one molecule of water is released. Tripeptide involves 3 of the amino acids, but 2 peptide bonds form, meaning 2 molecules of water is also released. So the best example of a tripeptide is glutathione. Glutathione, you'll know how important it is when we discuss metabolism in the pentose phosphate pathway, how it's used much in maintaining integrity of the red blood cells. That is the glutathione. It is a tripeptide which involves the glutamate, cysteine, and glycine. So it's going to form a tripeptide. 2 water molecules are going to be released. It's going to form 2 of the peptide bonds. So what you can see here, I was trying to draw it myself to show how a dipeptide looks like. And for example, I gave alanine and serine. So if you see such a dipeptide, it means draw these amino acids separately. I drew alanine here. You can see alanine, carboxyl terminal end, amino terminal end. The R side chain of alanine is CH3. Remember, serine, carboxyl terminal end, amino terminal end. The R side chain is CH2OH. The most important thing is draw the R side chain in this position, because if you put the R side chain vertically or horizontally, if you put them horizontally, not vertical, maybe here, you'll get confused on how to draw a peptide bond. Just put them, they can either be here or up here. Then you can bring the hydrogen down. Because in the formation of the peptide bond, if you put the R side chain comfortably down here, it's easier to draw the peptide bond. And I've mentioned here the OH, carboxyl terminal end of alanine, has to give us the OH, which is released. The hydrogen will come at the amino terminal end of the serine. So the carboxyl terminal end and the amino terminal end will attach themselves, and we have a peptide bond forming there and release of water. So the carboxyl terminal end of alanine attaches itself with the amino terminal end of serine, and then it forms a peptide bond. So we talk about a dipeptide. One molecule of water is released, but then we have the amino terminal end, we have the carboxyl terminal end of alanine attaches itself to the amino terminal end of serine. At this point is where a peptide bond is going to form. And then I've written down here, you have to add plus 1 molecule of water. So if I am to mark this question, I'll give this question 3 marks, because the first mark I'm trying to assess, can you tell the 1-letter code of those amino acid? A is what? You draw the correct structure, 1 mark. This one, serine. Draw serine nicely, 2nd mark. And it has to follow that pattern, AS. Don't arrange it in your own way. Since you can remember serine well, you bring serine first and then A. No, it is not SA, it is AS. You have to put it the way it is. It is us, no South Africa of SA. No, put it AS. And then draw them correctly. I'll give you 1 mark for each of the individual structures of amino acid. Then lastly, I give my half a mark for the dipeptide forming and another half a mark for water. So you'll have 3 marks. But if you don't give me individual amino acids and then you go straight and draw this one, maybe you got the structure of serine wrong, then everything will be wrong. You'll get a 0. But if you draw the individual amino acids, even if you get serine wrong, it means as much as you got the serine wrong, you'll get 1 mark, because the other wrong mark will go for serine. It means this one is wrong. Even your water is wrong. So it is good to give the structures of the individual amino acids so that you can earn a mark or 2. than getting none if you go straight and draw the peptide. So we can practice much. We can practice much by drawing any of these ones. But the point here is clear. If you have to draw any of the peptides, be it a tripeptide, be it a tetrapeptide, be it a pentapeptide, just remember to draw each of those amino acids separately and try to correctly remember the one letter code of the amino acids, the correct R side chain. Draw it nicely, you will get all your marks. The peptides are the ones which lead to an amino acid. So if you talk about a protein, they are united through a peptide bond. So in that case, you will be able to tell that proteins are polymers of amino acid. So if you have a length of those amino acids of which you can't count, you talk about a polypeptide. And that is why most of those proteins, their properties relate well with the components, amino acid components in them. So that one brings us to the final discussion on proteins or amino acids and peptides. So I'll next introduce fatty acids, which are the building blocks of lipids.